Okay, we are back. We just finished talking about DNA replication, copying that whole chromosome and all of the chromosomes, the whole genome. But now we're talking about how just one gene gets copied so that that gene can be used, right? You just drank some milk, your body needs to make the enzyme to digest that, right? An enzyme is a protein. You are doing something scary, your heart is pumping faster, your body needs some adrenaline, right? Um, these are when your body's not copying your whole genome when you need adrenaline, it's just copying the gene for adrenaline, right? So this process is different, just copying one little gene so that your body can then use that to make a protein. So that's what we're going to be talking about now. Now, the phases of making protein, first, it's in two steps. First is called transcription. That's copying the information from the DNA and making a piece of messenger RNA from it. And you can think of, this comes from the word for a scribe. A scribe is someone who writes, okay? And I want to see if I can get a smaller tip here on my pencil thing. Okay. Um, so you make this little piece of messenger RNA in transcription, and translation is taking that piece of messenger RNA and decoding it and turning it into a protein. Okay, so this is our decoding step. Okay, so we'll start with transcription. How does that work? Well, we've got this whole genome inside of every cell, right? And we need to figure out, okay, we just want to copy a little part of it. So the first step is that we talked about DNA polymerase when we were talking about DNA replication. Now we've got something else called RNA polymerase. This enzyme needs to recognize what gene we need to copy. Okay, we want adrenaline, find the adrenaline gene. So it goes and finds this promoter site. Kind of like a party promoter, right? They're like, come here, this is the spot you want. Okay, so it recognizes that. It's like, this is the start, whatever. And the RNA polymerase then unwinds just that little bit of DNA. It does not need to unwind the whole strand. It's not doing the DNA helicase job. It's just unwinding a tiny bit of DNA just for that gene. The polymerase then makes a copy of just that section of DNA. So if this was your DNA strand, the polymerase is just making a copy of that, and it's just making a single-stranded copy, right? So if it's got this piece of DNA, it's just making one little copy off of this, right? DNA that we're copying, this would be what it is, right? And then this is your messenger RNA. But the difference here that we need to keep straight between DNA and RNA is that RNA has no T's. So it has A and G and C, but instead of T's, it has U's. So I have to throw out what I just wrote here, and I've got it written in here. This is actually our mRNA translation. Anywhere there's an A where we would have matched it on DNA to be a T, now it's a U. So here we've got UUU. If there's a G, it's a C. If there's a T, it's an A. If there's a C, there's a G. If there's a G, there's a C, right? So it's just like making a copy of DNA from DNA, except instead of T's, you're going to put U's down. So this is immediately one difference between RNA and DNA is that they have one different base. Okay, so we've made this messenger RNA copy from our DNA. And we can then use that. So it's got this piece of DNA that it's copied. It's made this piece of RNA from it. Well, now we need to stop, right? So that RNA polymerase that was making the copy, it reaches something that says stop, you're done, this gene is over. It detaches from the DNA. The DNA is then going to reattach to itself, right? So we'd had our piece of DNA that got kind of separated, right? It was this double-stranded helix. We made, gosh, I'm having trouble switching colors because it's changed everything on me here. Okay, so here's our little piece of RNA copied from that. This RNA is then going to go off here. It's going to get edited. It hit the stop, right? This said start, go. This says stop. It then moves on to this next phase. We've just got this piece of messenger RNA. Our DNA is then going to get re-smushed together. Okay, it's not. It's as if nothing had happened to it. We've got this piece of messenger RNA. It's going to get edited. Make sure there's no mistakes. It's going to have a little like, you not need to finish it off. Otherwise, it's like a piece of, a shoelace without that little cap. It's just going to make a mess of things, right? So it gets capped, it gets edited, it's ready to move on. So all of this takes place in the nucleus because that's where the DNA is, right? But where are proteins made? Think about that for a sec, way back to chapter 2. Where did we make proteins? Proteins are made on the ribosomes, right? And the ribosomes are on the rough ER. So if we think back to what a cell looks like, right, here's our nucleus, and then outside the nucleus is the endoplasmic reticulum. Very convenient, right? This is all set up to be a really nice assembly line. So your messenger RNA leaves the nucleus and it goes out and it finds a ribosome to attach to on the rough ER so that a protein can get made. So our messenger RNA now leaves. Just to show you guys, before we move on to the next step, we've got this RNA. What is the difference between RNA and DNA, and what's the same? Okay, this is RNA on the left, DNA on the right. RNA, single strand. DNA, double strand. Another difference here, this has U's, uracil, this is T, thymine. So there's one different base here. They're showing you that, right? They're both just one ring, right? But they're different ones. There's another difference here is also the sugar. So this sugar is ribose for RNA. For DNA, the sugar on that backbone is deoxyribose. It's a ribose that's missing one oxygen. Okay, so it's a different sugar in the backbone, one different base. RNA is single-stranded, DNA is double-stranded. And there are different types of RNA. The one that we're talking about right now is messenger RNA, and that's probably the one you guys are most familiar with because that's what the vaccines have been made out of, but the Pfizer and the Moderna COVID vaccines. And when we finish talking about messenger RNA and transcription and translation, you will then understand how these vaccines work and why they're so effective. Okay, so this is our structure of RNA. It was made in the nucleus off of that DNA. It now leaves this. It's to go copied and turned into a protein. Okay. But first I'm going to show you some more pictures of transcription. We've already talked about this, right? So we've got our DNA template. That's this blue one. One of the strands is, that second strand of DNA. It's not getting copied, right? The RNA is only copying one side because it's single-stranded. So we've got our RNA here in pink getting copied off of the DNA. It had this promoter site that said start here, and it did, and it just keeps going until it hits the end, okay? And here it is, another picture. You've got your RNA strand getting made, and this big kind of blob in the background. This is your RNA polymerase, okay? It's just kind of moving on down this line of DNA, making the messenger RNA as it goes. And again, it's made from the 5' end to the 3'. So this thing strand of messenger RNA, and as the RNA polymerase leaves the DNA, reattaches itself to its complementary strand, and then winds itself up into a helix again. Okay, so this is transcription. The messenger RNA is then going to leave the nucleus. Translation is turning that messenger RNA into a protein. This is occurring in the cytoplasm where the ribosomes are. Also, floating around in the cytoplasm are amino acids, because remember, if this is turning into a protein is a string of amino acids, right? So this is our protein. These are amino acids held together by peptide bonds, right? So there are these amino acids floating around in the cytoplasm, and those can get plucked out. Oh, I need this one, I'll grab it. I need this other amino acid, I'll grab that. There are a lot of amino acids, right? And depending on the order they get strung together, it makes a different protein. So that messenger RNA finds a ribosome and it attaches to that, and that's where the protein is going to get made. Another piece of RNA comes along called the transfer RNA, and this is our translator. Okay, it's like the UN translator, translating from RNA code into amino acid code. Okay, it says, oh, there's a U here, a UAG. I know what that is, and it pulls out the right amino acid. Okay, so just the steps in this translation are, first of all, that messenger RNA goes and finds a ribosome. That ribosome is going to find the start, right? So remember, it needs to know which end to start at. There's a start. Every start of every protein that we know of is AUG. Every single one. So that promoter site in the DNA, it had something that was, it has to match this, right? It was TAC, and the complement to that is AUG. And if you've got an AUG, that means start here, okay? And that amino acid for every single protein out there, if there's an AUG, that's the amino acid methionine. So these are read in groups of 3, okay? Groups of 3 nucleic acids. Each of these bases, right? 3 bases. And those 3 bases, let me clear this. 3 bases, it is 1 amino acid. And those 3 bases are called a codon, right? So the start codon is AUG. Then you read the next 3, and that codes for 1 amino acid. And then you read the next 3. So it's not a 1-to-1 thing here. It's 3 nucleic bases is 1 amino acid. So the first step is you just recognize start here. It starts, it puts down methionine. And then that transfer RNA is going to run along the messenger RNA and read it, right? And figure out which is the next amino acid that you need. And then you get to a stop, and it says stop, we're done. And it's going to stop making that protein. That protein, then, remember, we talked about how amino acids have this primary structure, which is just the order of them. And then it has a secondary with hydrogen bonds folding it up, and then a tertiary. So then it gets all folded up and forms whatever shape it needs. We now still have that messenger RNA that was its template, right? That messenger RNA can be used again. So if your body needs more adrenaline, you can take that. It doesn't need to keep copying that strand of DNA and that gene from the DNA. It's made that messenger RNA. It can then get reused and make more adrenaline off of that, right? So it can get reused, reused, reused. And when it doesn't need it anymore, when your body says, I'm done, right? Eventually that adrenaline rush stops. It can then take apart that messenger RNA and just recycle the bits and use them again for something else. So we've got a picture here showing you, okay, so this big blue thing in the background is our ribosome. Okay, so here's our ribosome. It has actually 2 subunits. So we have this little subunit and we've got the big subunit. This purple thing is our messenger RNA. This was copied off of the DNA, right? And then you've got this tRNA that reads, as you see, it kind of looks like a cow udder, right? It's got 3 little parts. Here, it's reading what each 3 bases on this messenger RNA. And it says, okay, it says like U, I know what amino acid I need, and it plops it down. And then it reads the next 3 and it says, oh, that's UCU. That's a different amino acid. I'm going to plop that one down. And it just keeps building them as it goes. So it's just growing this chain. Now, there is a decoder ring that we have for knowing, based on what the DNA says, what the messenger RNA says, which amino acid is going to get put down. So we read this by looking, our first letter is on this left side here. Our second letter is along the top, and then the third letter is along the right. So we can read this if we look. If our first letter is U, our second and our third letter is, we get U, that is going to be read as phenylalanine. That's the amino acid that's going to put down. And there's some redundancy in this code. So if it says UUC, that's also going to read as phenylalanine. And you can see here there's just one start, that's this methionine along the bottom. It goes A, and then U, and then G. So you've got that here. Here's our start. Everybody starts the same. And then there are a few different things that code for stop. So it could be UAA, UAG, UGA. Those all mean stop. You guys do not need to memorize this chart, but you should know how to read it. So if you are told that there's something that says CCU, you should be able to look on this chart and figure out what amino acid would get put down. And you don't need to know the full name. You just look at CCU and you say, oh, well, that's pro, right? That's going to be that amino acid. And if it says CGU, oh, well, that's going to be this amino acid, right? So you should be able to decode from this chart. Now, again, remember these 3 in a row, these are called codons. This is the code. And using this chart, you can decode it and know what amino acid it's coding for. So here's just another picture of this process. We have our ribosomal subunits here. The messenger RNA attaches to it. That's this top picture. And you have this tRNA that figures out which one it matches to, right? So it starts with AUG, it puts down methionine. Another tRNA comes along, it says, oh, it's UUU, we're going to give you this PHE, right? And you can see as you look here, from the left, the picture on the left to the chart on the right, you can see how these things are matching up. So it just starts to form this long chain. So now we have all of these amino acids forming. Now you can see or you know that your body isn't always making every single protein all of the time. You're not always making adrenaline. A baby has its full genome in all of its cells, but it has not yet gone through puberty, right? It doesn't have all of those hormones being made. Bodies make things when they need them and they don't make them. And there are things that turn on genes when they're needed, whether that's an environmental cue or something in an internal cue. Different genes are being made at different times or being copied at different times. So this is gene regulation, right? So your gene expression is not just that you have that gene, but it's also that gene getting turned on or off. You're not always making the gene for digesting milk. You're making that after you drank a milk product, right? Or ate a milk product. Your body didn't start making hormones until you reached a certain age, and then it started making those. Your body didn't start growing, right? I mean, babies are growing, but you hit these certain growth spurts where you get really bigger, and then those turn off. You don't grow throughout your whole life, right? So that messenger RNA is made only when a gene is turned on. And there are certain ways you can study it to see. This is a microarray, this picture at the bottom, and it shows you could look at a genome and see which genes are getting turned on or are off at a specific time. And you can compare them between individuals and see, okay, you know, when they're exposed to this thing, everybody makes this gene suddenly. And then we can pinpoint where that gene is. Or you can do twin studies, and this has been done with the Kellys, who are astronauts. So there's Mark Kelly and his brother, I think Scott Kelly, and one went in, they've both been in space, but one spent like 6 months in space, and while one was on the ground. And then when he came home, they could compare their genes. So, and which genes were being expressed at the time. So this is how that messenger RNA is used to make genes. And I think we're going to stop at this point, and we will pick up where we left off.